Hello, Nithya. Thank you for joining us today.
Hi, Nico. How are you?
I'm great. How was the weather over in Boston?
The weather's okay. It's 60 degrees right now, so I'm just looking forward to sitting outside for baseball and softball later tonight. It's going to get colder.
Right. Yeah, it's about the same here in Montreal. But yeah, for our listeners today, Nithya, you are a experienced CDMO veteran with both experience on the technical and business development side. And today, I'm excited to talk with you about the sort of communication challenges, both in the technical aspects and in the business aspects of the interactions between CDMOs and biotech companies that hire out to them. So, something that we've previously discussed that could be a good starting point was in the market, the post pandemic shift from comprehensive development in CDMOs to minimal, good enough for phase one approaches.
Yeah, so over the pandemic, one of the main reasons I think a lot of CDMOs started popping up is just the general news of vaccines and medicines and the supply chain shortages. They weren't reaching patients. Even in our daily consumer driven lives, we had challenges with general cleaning supplies, paper supplies, and even food shortages. So I think everyone was trying to figure out how do we solve these supply chain challenges. So CDMOs popped up. And it was great. Now that we weren't having any challenges for cell therapies, gene therapies, biologics, what have you, there was a great need for that. But what's happening is that I think there's too much going on. A lot of people were able to raise the funds and open up those CDMOs and showcase how they're going to be different. But as we started normalizing the situation, everyone realized, hey, maybe we hired too quickly, we purchased too quickly, too soon. It's normalizing out to what we actually need and how we need to increase the supplies and medicines to the patients. And we're normalizing the price tag as well. Back a few years ago, when I actually joined Resilience, we were meeting customers we needed to educate them. There was the need for them to go from start to finish. They didn't know how they were going to get there. So we were able to provide them with guidance. We said, do X, Y, and Z, and we'll get you the best product to the patients. But as the market changed, now our customers were saying, look, what you provide us is great, but we can't afford to do all the bells and whistles for process development into manufacturing. The question started becoming, what can you do to support us with the finite amount of cash that we do have to ensure we can get to at least phase one clinical trials? Because a lot of our clients are small biotech, and what they're trying to do is meet certain milestones and goals so they can unlock more funding so they can further their therapy into the clinic. So CDMOs, including Resilience, what we started doing is evolving the offering, evolving what we can do for the client, and really providing them with a minimum viable process that they can utilize to ensure that they get their IND filed and accepted and be able to move to phase one clinical trials. And that's the market shift. We used to be able to say we can do everything and give you the best de-risked process, but we had to start educating our clients and saying we potentially could take away this step to help accelerate the process, development into manufacturing and reducing costs. But it's at the end of the day, it's a decision that we ask the client to make because we want to make sure that they are educated, providing them enough data to say this is the pros and cons of taking away certain steps, but if you're willing to take on the risk and we mutually agree, we'll move forward.
Yeah, and so through these conversations, one of these risks that's come up has been engineering runs. So it'd be great to discuss a little bit about the sort of risks and opportunities of accelerating the process of tech transfer and getting to production and the potential savings of not doing things like engineering runs. What are some of the other factors that companies will consider or may not want to engage with to save costs?
So to save costs, the CDMO is always going, when they come up with a new platform, they're going to want to sell this to a first few customers to see how we can move that into the GMP space. But the opposite end of it, these customers don't want to be the first. So they don't want to be the first to run a process. They expect the CDMO to already have run some engineering runs in their GMP space to showcase confidence. So it's always that balance of what you're showing in terms of experience and new platforms. If there are platforms that you've run 20, 30 times and more than 95% successful, then there's an opportunity for you to let the customer know, look, this is how much experience we've had, this is our success rate, and providing that information and data will allow both parties to mutually agree whether we should move forward with an engineering run. Now, if it's a new platform, new process, it's a completely new modality, and now you're foregoing those engineering runs either from the CDMO side or the customer side, it's a higher risk. You start failing your engineering runs, not only have you wasted time and money, but there potentially may not be any way to move to the GMP run because you may have to go back to the drawing board and figure out where the error came from that engineering run. Sometimes it's very simple, sometimes it's not. And for some of these therapies, they rely on long lead times for the raw materials as well. When it comes to gene therapies, relying on these plasmids, right? So if we don't have a strategic partner to source these plasmids who's willing to quickly manufacture them for you, or you didn't account for an error and you only purchased enough for one batch, you are now waiting, depending on where you are in the queue with your partner, maybe 4 to 5 months or 7 to 9 months, or maybe more than a year to get more plasmids for your run. So it's very important to plan for those risks. If you're going to forego an engineering run, make sure that the CDMO and the partner, we're documenting the risks associated with this and where you see those risks are. So that risk analysis, cost analysis is so important in making that decision. And same thing the other way is that if you are going to do an engineering run and suppose it's going to fail, we always want to plan for the negative. We're scientists. We also want to plan for the failure, right? As much as we always want to say we're going to be successful, plan for those extra cost of plasmids, plan for working with your partners and having something in backup. Worst case, those plasmids, you use them for another batch, right? So plan accordingly that you have a use for those plasmids and you're not just purchasing something that's going to be wasted on the shelf and you now spent $1 million on. So I think it's more important than making a quick decision on the fly. It's really having those conversations across the stakeholders to really document the risks, the challenges, and the mitigation plans if something goes wrong, and see from the biotech side, see how this now balances off with your costs as well. Can you risk spending this money with an engineering run or not an engineering run and still be successful? I think CDMOs, we can be your consultants, but we can't make the final decision when you're from a CDMO. The decision ultimately needs to come from the small biotech to see what they're willing to stomach based off of the data that the CDMO or the experts providing you.
Okay, so it sounds like there really is a huge difference in that risk of the engineering run based on the sort of level of innovation and novelty of that technology. If you're using an existing modality that the CDMO has run many times, you can expect that process is going to work. And it sounds part of what you get out of the engineering run, aside from just knowing that the sort of biochemistry is working as expected, you're also testing the logistics. Like you mentioned, having the plasmids on hand. And is that something that might be taken for granted sometimes?
I believe so. When these timelines are put together from a CDMO perspective, there's always these assumptions that are put in there. It's assuming all raw materials, all testing, and everything is available, even capacity. But in reality, there's going to be delays to your project start if the plasmids and other raw materials, even bags or connectors, whatever you need from the process side isn't available. There's going to be delays if there's no capacity. We're assuming there's infinite capacity in our CDMOs when we present and sell to a customer. But I'm sure a customer realizes in reality, it's not infinite capacity. So it's making sure that you are accounting for any delays associated with capacity restrictions. And then, of course, you're also accounting for anything that might be just from a general logistics standpoint, holidays, downtime, equipment maintenance, all of those aspects, once again, are not typically considered during the selling process, but it's very important from a biotech side and the CDMO side. CDMOs are not trying to be deceptive. This is just the reality and how typically offerings are sold. It's basically a proposal with 20, 30 plus different assumptions. And one of the assumptions goes out the door, there are delays. Not trying to be deceptive, everything's on paper, but biotech to make sure that you understand those assumptions and understand those challenges when you go present and say, hey, we're saying we're going to get material in 12 months. Could it be 12 months? It could be less. It could be more, depending on what stage of all aspects of the process are.
Okay, so yeah, everything in those sort of tech transfers has to happen as expected in order to get that timeline that you'd expect.
Yep.
So one thing we've touched on before is the methods, the sort of cell culture methods, equipment compatibility, some of those logistics. And you mentioned, so we've discussed the sort of differences based on the novelty of the technology. And I'm curious if during the tech transfer process, if you might see more of that back and forth communication to ensure alignment of methods, whether they be like process methods, analytical methods, with those novel technologies compared to existing modalities.
Absolutely. The best CDMOs, I think, out there really spend a lot of effort to ensure their tech transfers are successful. New equipment, existing equipment, new methods, existing method, everything. And typically, just my experience with resilience and potentially, I think this is happening to other CDMOs too, is there is a pilot lab or a demo lab that exists in the development space. So it is at a certain scale. My experience is having it maybe in the gene therapy space or biologic space, having it up to a 200-liter demo bioreactor in the space allows for you to mimic the process that could happen at the GMP space. How to de-risk those tech transfers is ensuring the equipment that's in the demo space mimics what's out there in the GMP space. Of course, depending on how you're scaling up in the GMP space, there could be some mismatches, but that's where engineering comes in, right? Now, there's a reason there's dimensionless numbers and engineering principles to help you scale up appropriately from the 50 or 200 liters in the demo lab up to the 500 up to 2,000 in a GMP space. So engineering principles, we'll trust them and we're going to go with them. But in general, the equipment for analytics, process, the TFFs, whatever have you equipment are going to be as mimic as closely as possible. And more importantly, you can't just rely on the equipment and the tech transfer via, hey, I did the parameter here versus there, but it's bringing your MSAT team and maybe other operators from your GMP space into the demo lab space, into your pilot lab, and really observing how things are being operated. You and I have both talked about this, and we really mentioned that one little thing, regardless of how much it's written, can make a difference in how you run a process. So sometimes those little tweaks or inadvertent touch of a knob or a tube might make a difference, and you don't capture that when you're tech transferring the process. So ensuring that you're bringing your MSAT team or your operators in to take a look. At the same point, when you're doing your engineering run or your GMP run, depending on how the client and the company decide to move forward, that your pilot lab or demo lab team are actually going down to your GMP space. And for the very first run, working with your operators and MSAT team to ensure success of that first run. You could have your GMP suite just on the other side of the wall of your demo lab, and you could still have challenges. But you're now talking about multiple CDMOs having multiple sites across North America and the world, and your development lab might be in North America, but your manufacturing might be in Europe. How do you ensure the transfer? So it could be around the corner, it could be across the sea in another country. It's all important to make sure there's communication and ensuring the general human error is reduced. I said you could do everything via the computer, AI, and other technologies, but there's still a level of human nature that needs to come in to help verify what you have makes sense. You've always heard, don't just trust everything on ChatGPT, right? Go and verify what you have on there, tweak it. And that's the same thing. Technology is great. AI-based methods are great. Even Excel, whatever have you is great. But there's still room for human error. So it's very important to go in and double check so that your tech transfer can be successful.
Yeah, that makes a lot of sense. And it really comes down, it seems like it really does come down to a communication issue. And so one thing that I've noticed is that these communication issues, they tend to be less of a problem when the two parties are less siloed. So within a CDMO, for instance, when you have a pilot plant and a GMP plant, and they are using the same documentation, same language, they understand each other's processes very well. That doesn't seem to be it. The communication generally seems to flow quite well in those circumstances. My understanding is that it becomes more challenging moving from the biotech into the pilot, because up until that point, these two companies have been operating siloed. Is that your perspective?
There's definitely a difference in what someone thinks a tech transfer is, right? CDMOs generally are very good. They have checklists. They understand what they need to ensure the proper tech transfer of client process or construct or information into our database and systems to show that we have a successful tech transfer. But we've had experiences where certain companies believe just writing something down on a PowerPoint is a tech transfer. And we said no. And to be honest, I think that's what helped guide us to say, let's put together an actual checklist, a true checklist, so that we can help educate the client on what they need to provide to us. I think more often than not, CDMOs know what they're looking for, and we assume the clients do, but it's just an education. A lot of the clients could be 2-person companies, 10-person companies, they could be virtual companies, or they just have something on the bench, right? And they're not experienced in how to interact with manufacturers. They're going to manufacturers because manufacturers are the experts. They know what they need to do. So I think it's very important to go back to the customer and saying, hey, if you don't have these certain documents or this information for us, we may have to go back a few steps so that we can get to where we need to be successful. And once again, we're looking at the CDMOs as experts. They're the consultants. We need to know this is what you need to be successful and educate the small biotech. Our large pharma counterparts, they probably have more experience in what they're looking for, because more often than not, large pharma have their own manufacturing. So they know how they're transferring their processes in. When large pharma comes in, they're looking for additional manufacturing capacity. They're probably in their later phases. They're probably pivotal, almost commercial. They're looking for secondary manufacturers. So those tech transfers tend to go more smoothly. Not saying always smooth, but more smoothly. But when you're going to a smaller segment, like in a smaller biotech, sometimes they're spun out of academia. I was in academia. I know exactly what it means to transfer a protocol from me to a colleague. I write in 10 lines. That's pretty much not what you need for a CDMO. It's very important for us to just educate and work with them, not get frustrated. I think that's what happens a lot of times, is both sides can get frustrated. It's more so make sure we're partners in this. Let's work together and let's find ways to become successful. At the end of the day, we're all working for the patient. We're trying to give the best opportunity for that patient to lead a better life.
Of course, yeah, it makes total sense. And yeah, you mentioned the translation from academic or sort of CRO discovery labs. And something that's come up a few times is the sort of differences in expectations around documentation. So you mentioned a protocol, and sometimes there are assumptions about that a protocol might translate directly to what an SOP is. And something that I think is missing is there are operational details in SOPs about how this is happening in this circumstance. And I've also heard that I've noticed there seems to be a general trend in the documentation where the sort of first tech transfer you talked about from the academic or discovery lab to process development, that is a work of characterization, of getting details hammered down. And then moving from the CDMO to a manufacturer, it seems like most of those details have been figured out at that point. But it seems that there's this general flow of sort of the least details being at the earlier stages, and then through this sort of life cycle, getting better characterized. So yeah, I'd love to hear about that sort of process from your perspective.
Absolutely. And I'll give you a couple examples from my own experience from being in the scientific lab. I'll give academic and industry, right? So in academia, part of my thesis was to transplant Schwann cells. So Schwann cells are the cells that myelinate the peripheral neurons. They help transmit the signals from the brain all the way to your extremities, so, your arms, your legs. If you have an injury, your nerves are going to demyelinate, so you actually don't get those functions working appropriately. So I was transplanting Schwann cells into acellular nerve grafts. So these grafts are from donor nerves. They've been decellularized, but they have the extracellular matrix. And me and my counterpart in the Department of Plastic Surgery, we said, okay, let's try to do this project. And so we started putting the Schwann cells under the epineurium of this graft, and the Schwann cells kept flowing out. And we were following protocols that were published in papers. And if you read papers, they're not that detailed, right? So
Yeah.
sometimes they're successful, sometimes they're not. So what my counterpart and I did is that we completed a whole study to take a look at different size of needle gauges using Hamilton syringes and really figured out how we can deploy viable cells and transplant viable cells into these grafts. And we actually labeled them with Q tracker, so Q dots, so that we could look at them later and see how many cells were actually remaining. And we ended up publishing a paper on this so that other people can actually utilize this method to be more successful in transplanting Schwann cells into these acellular nerve grafts to drive neuroregeneration. So it went from us being failures, going after a couple of papers, is like, let's actually write a paper here and showcase how to do this. And then we can utilize that same method every time we do some transplantation. And we were able to do that for subsequent papers and projects that we did. So it's very important from the academic field. A lot of these methods and material descriptions you have aren't that clear. So you're pretty much trying to figure out a lot more details and figuring out how to actually be successful. And especially a PhD student, you want to be successful. You don't want to be there in grad school for years. You want to get out in a reasonable time so that we can pursue the next parts of our career. In industry, where I found this I found it funny actually, and was, in all seriousness, it was good for me to learn these protocols. I was learning how to do Western blots in my first job. And my boss wrote these protocols on a paper towel. He pretty much do this. He wrote like numbers and everything. I was like, okay, here's a paper towel. I'm going to learn by observing him and how he's doing these Western blots. And I think learning from the very basics of doing Western blots, I became very successful at doing them and understanding where to troubleshoot, if your buffer's wrong, and so on and so forth. But here's the challenge. We were getting some colleagues from Japan. And my boss had told me, look, we need to write down all our protocols so that in a book, like SOPs, so that we hand it to our Japanese colleagues.
From the paper towel, that you need a book out of the paper towel.
Yeah,
now.
so paper towel to my brain and now writing it down on a piece of paper. I had a lab partner at that point. So she and I spent probably a couple of weeks writing down all the protocols for Western blots, passaging, qPCR, everything, all protocols that we did, and making sure it made sense in our heads. But that's the problem. It makes sense to you because you always do it. So it's very fortunate for us when our colleagues from Japan came, handed them the book of protocols and then, hey, this doesn't make sense, that doesn't make sense. And we were able to refine them, eventually being able to make those more standardized procedures for any new scientist that came into our lab. But these are the experiences that you have not only in academia, but you have them in more development labs within industry as well, because it's a bunch of scientists, which speed is the game as you're trying to screen through whatever compounds or RNAs or molecules that you are. You need to keep going. You never think about the little details that can make a difference.
Absolutely. That makes a lot of sense. So during the story you just mentioned about your graduate research, like you wrote basically a methods paper of how to do this procedure. And something that came to mind is that in academic research, I'm not sure if you agree, but sometimes I feel that there is a sort of premium for results and that the methods might not always be as, like scientists don't always look straight to the methods when they're interested or seeing new research. Often they'll be like, wow, I want to see the pictures, the figures that are in the results. And I wonder if this sort of culture might be part of the challenge during these tech transfers, where through academic training in PhDs and graduate programs in general, scientists are reading papers and looking at the results, and the methods can sometimes be boring because it's not as, it doesn't really tell the story as well. So I wonder if in your eyes that might be part of where this sort of communication challenge might come from.
No, I tend to agree with that actually, because as a scientist, when I read papers, you go to the results in the discussion section, you go to abstract results discussion. A lot of papers actually will put your methods and materials at the end. A lot of times they're not there. Half of them might be in supplemental material. So they're not actually deemed as like the most important part of the paper. But when you're looking in academia or when you're looking for a new method to develop in industry, you are going to go to those methods papers and those sections, and you're going to find incomplete information. So you're going to spend time developing. But to your point, this is probably where a lot of it stems from. I'm not saying everyone is like this. A lot of people did come from academia, whether they did work in undergrad through grad school. So that's the general idea when you're sitting in small biotech, large pharma, you're more in these discovery research labs that are moving it through. Now, the CDMO world requires a different mindset. It requires more systematic approaches. It requires more attention to details. unless you are part of a group that's developing new methods, most of these people are more scientists that really believe that work in the step-by-step method, the details, engineers, very different sort of mindset. So if you're coming to a CDMO to do research, it's not the place for you because it's more a product-based goal, because at the end of the day, you're doing some sort of research and development, but it's for the movement of a product through manufacturing. It's not a primary discovery role. So the two aspects that I described in my career experience was in academia and more in the discovery lab. But working in a CDMO world, you're gonna have to think differently. But there's that switch that's needed to think more product-based than research-based. And I think you want to hire talent in CDMOs that are a lot of brilliant folks, a lot of great scientists, but a lot of the folks that work in CDMO world, they're very much that broad science knowledge. They might be coming from biotech and large pharma. And in my experience, you need a nice mix of people who have that more focused approach so that you can think about products rather than research. I often joke around that if people started developing their therapeutics the way a product manager would, we would probably have better outputs of our therapeutics. But the product life cycle of something that's on the shelf or instruments is probably anywhere from 6 months to 3 years. So it's an easier aspect as a product manager to plan for versus a therapeutic can be 10 plus years. So for you to plan for commercialization and everything that you need to do on day one may seem like, hey, that's 10 years away. I don't need to care about it right now.
Yeah, it makes a lot of sense. And one sort of phrase that's come up during this transition, and apparently this seems to happen more with younger companies, is the understanding that you really do want to do all of the R&D at the bench scale, and that it becomes incredibly expensive and inefficient to do large scale R&D. So yeah, I guess this is something that happens sometimes, is that being only in an R&D environment, you might not think about that because you haven't worked necessarily in like a manufacturing environment.
But that's where we need to rely on newer technologies that are being put out for these different therapies. Even if you're doing it on the bench, there are plenty of bioreactors and reagents or media that translate well into the manufacturing scale. There are scale down models, Sartorius bioreactors or ScaleReady's G-Rex or PBS Biotech's vertical wheel bioreactor, microfluidics. All of those reactors have meant to be scaled up, and you should be using the engineering principles to get there. Same thing with your reagents. If you start selecting your reagents from day one, that thinking you're going to be in a manufacturing environment, you'll select reagents that are probably animal-free, xeno-free, GMP grade, whatever that you need for your target therapeutic. So I think that's the important part. If you're looking to get a product to the clinic, as you translate it all the way from the benchtop to those larger scale bioreactors, that you're able to translate everything appropriately. And that makes it easier for the tech transfer, that makes it easier for the supply chain and just the partnerships that you need to keep going. And that's where calling all of our tech innovators, the space out there, when you're putting something out there, please think about how you want to translate this, not only from the R&D space, but all the way to the GMP space, making sure it's meeting regulatory and safety guidelines. I think that's the important part. And those are the partners and tech innovators that are going to be successful, because we're going to be able to decrease the risks of tech transfer, because you're not changing a reactor, you're not changing a reagent from this scale to that scale. You are now saying everything you do here translates all the way to larger scale, and it's a matter of making sure it goes smoother with all the agreements and partnerships you need in place.
Yeah, so it sounds like the R&D labs that will be successful are those that are really treating research as a scale down model of manufacturing.
Yeah, and that will help actually once you get to your manufacturing scale. Once you go beyond phase one, you're going to do your process characterization, right? If you already had something that you set up as a scale down model, your process characterization step may not take as long. Process characterization can be expensive when you're really trying to find your failure points. But if you treated your earlier stage as like really understanding at least some parameters in your design space, then, coming back when you're going into your pivotal and later phases, that you might be able to lessen your costs and go faster. And that's what I'm saying. When you're developing a product, like an off-the-shelf product, a SKU, so I was a product manager. I'm a product manager at heart. It's really spending your first few steps of your development thinking about the end goal. If you do that and put all that effort at the beginning, as you move forward to your manufacturing and commercialization and launch, you're gonna have more probability of success and less risk. The last thing you want is once you get to commercialization, if you didn't do this and you have to go back to the beginning, and now it takes more time, effort, money, resources, and you may not be able to commercialize your product because you can't raise the funds for that. So that's why I'm saying if we could treat every therapeutic development as the way you would treat a new product development, we might see more success. But that's really I won't say a difficult mindset, but that's just a mindset change to think more long term. And it's definitely harder when you're thinking 10 years ahead versus 2 to 3 years ahead and where you want your products to be.
Yeah, and you're using the word product, and it makes sense. And I wonder if maybe part of where that divide might come from, because that really is taking it like a product management perspective, a long-term view of, yeah, how can we do this in such a way that it's going to work at the end at scale? I wonder if also that some of these challenges might come from, if you come from an academic background and you're trained in research, I never had a product class in graduate school. Yeah, I think it's just in my eyes, it's a different perspective, and maybe not one you might think intuitively you need at the beginning stages of a biotech company.
Yeah, absolutely. I'm working with a company right now, and they have these off-the-shelf iPSC cells that have been edited to remove the HLA-1 and HLA-2, basically to make sure that what they're calling is immune cloak. And, when I'm trying to ask them, why is this important, right? And, what's great about the cell line, they told me the cell line was made in a very GMP compliant way. And they're trying to make it immune cloak. So now you can create allogeneic therapies, and they've already created these from donor cells. And why this is important is that if you were to create your own cells, choose your own donor and create your own cell line, you might be spending another 9 to 12 months creating that cell line. The other aspect is having these ready to go cell lines or these ready to go technologies prevents someone from starting in the wrong way. So once again, if they start with an R&D bank and doing all these edits, they may have to repeat the whole process to create the GMP compliant cell bank. So it's a matter of understanding what you want to do. If you're going to create an R&D cell bank and just get going, and for example, if you're in academia, you're working on developing novel therapies, looking at novel pathways, novel differentiation methods, that's okay. But at some point, if you want to translate that into something of a therapy, you're going to have to start from square one. So it's important to really think about your starting materials if you end up wanting to go into the clinic into commercialization. And that's why I say, think like you're making a product so that what you choose as being is important. I'll give you an example of something that we didn't think about when I was at Quad Technologies that was acquired by Biotech. We had a buffer that had BSA in it. And BSA, bovine serum albumin, can't really be used in clinical, any trace of it in clinical cell therapies. So we didn't think about when we had that product in R&D time that we would think about the end user, the clinical aspects of it, which is probably a mistake on our part. And second of all, we couldn't even ship that to Japan because Japan doesn't accept the BSA-based buffer. And this is all to say that regulatory agencies in different countries are different too. So something to think about. So we ended up doing is reformulating the buffer to utilize a recombinant human serum albumin made from rice. So we needed to find something that would still be as functional as the BSA, but also animal-free, xeno-free, so that we could claim that within our product so that cell therapy manufacturers would use this product for the expansion of their T cells. It's a lot to think about. Just these little aspects that you don't think are important are so important. And once again, that helps with the transfer into manufacturing. It helps with de-risking the process, and it helps with anything that needs to go downstream from regulatory safety perspective.
Okay. So something that just came to mind, thinking about the sort of translation from R&D downwards from that sort of product perspectives, I'm curious, is there a standardized way for R&D scientists to quickly and effectively know whether their process is going to work downstream and like in clinical trials? For example, so in that BSA instance. It sounds like that's right now, it's like an ad hoc. You find out when it's too late, maybe sometimes. And I wonder if there is a standardized way that R&D scientists might be able to, as they're developing a process, look and say, okay, is there like a bank or something like that where they can look to know if their process and the materials they're using are going to be usable downstream?
So there's three things I would say. One, don't work in a silo.
Yeah.
Talk to folks, network, talk to everyone, and make sure that everyone is in agreement how you're moving forward. Two, yeah, there are plenty of places for you to go and take a look, and you can go FDA guidelines, regulatory guidelines, reading the C of A's, the TSE, BSE statements, I think in like different products, like everything has statements, right? But it's very time consuming. So the best place for you to do is to collaborate with your regulatory and quality folks, right? As they say, when you don't work in a silo, engage with them early on. When making products, when making therapeutic products and saying this is how we need to proceed, work with them and make sure they sign off on the aspect that you're looking to incorporate into your therapy. And I think that's the important part. When I was at Biotech, from a product manager standpoint, that's one of the aspects that I was really trying to push. I actually designed and deployed a new product development process. And the first couple of steps was really engaging with your stakeholders to ensure that everyone is in alignment of this is how we're going to develop the product. We know which markets we're going into, which materials we may need to access. Is there any IP issues, challenges with these therapies? We need to make sure you're not being constricted on where the materials are coming from or your construct design. There's so much going on, IP issues. Can we manufacture this? Your supply chain. How are we going to commercialize this? It's really giving it a full idea of the roadmap and how we're going to move forward with the development with our other stakeholders. So I'm talking from a product standpoint, it's a lot easier to, once again, think about something that might come out in a few years, but there's some aspects of this that I believe that you can pull for an R&D scientist to really make sure they engage with the stakeholders that are going to be helping to push this product through. More importantly, your tech ops, manufacturing, quality, regulatory, to ensure you are developing under the right regulations and the guidelines, right? Because as I said, different countries have different regulations as well. So let's make sure if you want to commercialize in APAC or EMEA or North America, where you're going to go, that you're meeting those as well. The last thing you want is to reformulate a therapy to go into another market, because now you're just spending more money. But maybe that market actually has most people there that you want to send this therapy to. So once again, it's a lot of pre-planning. It's a lot of discussion ahead of time. Where are the challenges today on that? We don't have the time, right?
Yeah.
That's the problem. There was a luxury. There was a time at which you could have sat and had these conversations. But where the market is today, you don't have the luxury for those conversations. So who can help you there is your CDMOs. Is really taking a look at what you have, partnering with CDMOs early on, because they have all those people they can talk to instead of using an R&D person, trying to find the right regulatory person to talk to. CDMOs typically have those relationships. They have manufacturing, they have the legal. Let's help you. Let's take a look at what you have and give you some guidance on how we can continue the development path forward. So either you continue it and you bring it into the CDMO, or you bring the CDMO and we continue the development for you. I think that's what needs to get done. Is if biotech decides they're going to do everything on their own, it could go slower depending on their funding and their resources. So as I said, partnering with CDMOs and maybe even contractors earlier on can give you the proper guidance. But once again, we're still not at the luxury where you can have the full conversation. So it's having a more focused conversation that's going to get you through to phase one, but putting things in the parking lot for things that you need, and then going back to those that you need to address going into phase two. There may be certain methods that's okay if they're not validated going into phase one, but by the time you do your stability studies, they need to be validated. So I think it's having those conversations. It's a matter of understanding what grade of material you need for each of those phases as you move forward. Let's go back to plasmids. Like phase one, FDA says GMP plasmids are acceptable. I don't know if those regulations are changing. I haven't kept up to date on that, but at least until recently, that's what my understanding is. And then the later phases, you're going to move into the commercial grade plasmids. which adds costs, but at least you can understand that this grade is sufficient to get into your phase one. I think it's working with your regulatory agencies, your quality folks, saying what is acceptable for us to get this through to phase one and knowing what we need to do to bridge it to phase two.
That makes, total sense. And so it really sounds like it's like the solution to this sort of long-term planning challenge and problem is communication with all of those important stakeholders. And you mentioned quality teams. So one thing that I've heard is through the sort of development process of biotech companies, quality teams aren't always hired at the very beginning. And I understand that sort of through funding cycles, new stakeholders are brought on board. So I guess from your perspective, would you say that the CDMOs, an additional sort of value they bring is giving you some access to, for instance, those quality teams when you have those questions at that R&D level? For instance, the BSA example you gave, would that be a good sort of playbook to say, okay, we've got an R&D level process, now let's go to the CDMO, run it by their consultants to see if this is actually something we'll be able to move forward with before they have their own quality team?
Yeah, absolutely. So one of the things that Resilience was doing, and I believe they continue to do, so we had a really great regulatory team. So we would offer services separate from general CDMO services to help consult with the small biotech to see how we can bring their process into the clinic. So Resilience is doing this. I'm sure other companies are doing this as well, because most CDMOs will have that solid quality and regulatory teams. Because at the end of the day, your product needs to be safe as it gets to the patient, right? And you can manufacture all you want, but if it doesn't meet these two guidelines or the requirements of these two teams, it's not going anywhere. It's sitting in quarantine for a while. So most CDMOs will invest heavily on making sure their quality and regulatory teams are educated and up to date. They can support INDs, go with you for your pre-IND reviews, do whatever you need to make sure you're successful. At the end of the day, when you partner with the CDMO is successful only if the small biotech is successful. So we're going to work together. The best CDMOs are the ones that are going to make it partnership instead of transactional relationship. And I think those are the best CDMOs that will succeed, is when they actually work together rather than, hey, this is off the menu, we're done, pay us money and go.
Yeah, and that makes a lot of sense because, these are hopefully companies that will come back for more manufacturing down the line. Wow, we've covered a lot today. So I think one last question looking into the future would be, what would be your advice for new biotech companies that are starting in the next few months and they haven't hired everyone yet and they're at the company formation stage? What sort of advice might you give to them to improve their chances of success looking down the road towards manufacturing and then the clinic?
One big piece of advice that I learned from my whole development and commercial career so far is make decisions based on data. Don't make decisions based on a whim. Make sure that when you're deciding, hey, this is the construct or this is the molecule, from a science perspective, it makes sense that you're going in that direction. And if you're still not able to make that decision as you're forming your company, make sure you're reaching out to industry experts or academic KOLs, right? Your academic KOLs and industry experts are the ones that are going to help guide you to success. One thing that everyone wants you to succeed. And depending on who you are, your network might be great. You might be able to actually have a conversation over some coffee with someone to be like, hey, I'm thinking of doing this. Does it make sense? What else should I do? I think don't be afraid to talk to people and get some advice. I think a lot of times we want to hold technology close to our heart, but depending on how you proceed, I think having the right conversations are helpful. I've always noticed, especially when it's at Biotech and some other companies, having those silos up prevents success. And once you start breaking them and building conduits across business units, across teams, and across the company in general, and being more transparent and communicative, more success.
That makes a lot of sense. And there's a parallel in software that all of the money is in the integration. Everybody wins when people are better integrated. Yeah.
Absolutely. And it comes from any company, stems from the culture, stems from the leadership, stems from how they want to bring down and do that. Companies that push for collaboration, cross-functional leadership, matrix organizations, I'm using a lot of buzzwords here, but all these buzzwords are there for a reason, is that they want to make sure that everyone is integrated and you're integrating technologies appropriately, because you win, everyone wins. One failure, the whole company loses. And in a market like today, everyone needs a win.
Yeah. Wow. Thank you so much for joining us today. It's been a super informative discussion. And yeah, I'll end the recording here. And yeah, it was great.
Thanks, Nico. Yeah, no, this was great. Thanks for having me on the podcast. It's pretty great to have a conversation like this. So I really appreciate it.